Hello, my name is Dr. Susanna Lopez-Legentil, and today I'm here to talk with you about Ascidians or sea squirts right here behind me. Ready? Let's see together why it is important that we learn more about Ascidians. Ascidians are classified within the Phylum Chordata, just like you and I, and are our closest invertebrate relatives. Animals from the subphylum Tunicata are called tunicates and include over 3,500 species, and they're all marine. So we can find them in the Med or in the Antarctica or in the Atlantic. Wherever there is seawater, there is tunicates. Tunicates live in all seas then, from the shoreline to the great depths. The name tunicate describes the tunic, this yellow thing that you can see in this picture that surrounds the animal, and is made of tunicine, pretty much like the cellulose of the plants. The consistency of the tunic varies enormously. You can have a slimy, soft tunic, or a hard like a rock, leather-like tunic. There are three classes or big groups of tunicates. First, we have the class Taliacea, and those are mostly free-living animals that are nearly transparent and live in the water column. It includes what we know as pyrosomes and salps. Then there is the class Appendicularia that are encountered in most areas of the ocean and are very teeny, 5 and 6 millimeters in length. And finally, we have the class Ascidiacea, or the Ascidians. Ascidians are also called sea squirts because if you press their body, they will squirt at you. The class Ascidiacea is by far the biggest in the tunicates, with over 3,000 species described. Adults are sessile. That means that they cannot move. They'll just live in the same spot all their life, and they can attach to all kinds of substrates like floating dogs or rocks or other animals. They're often misidentified as sponges. Quiz time! If I ask you to tell me which of those two is a sponge and which one is a sea squirt or an ascidian, what would you guess? Tick tock, tick tock. Is the sponge the one in the left? Tick tock, tick tock. Is the one the sponge in the right? And the correct answer is the ascidian is in your right. So as you can see, those can be quite difficult to identify. In fact, several Ascidian species have worldwide distributions because on top, they are really good hitchhikers. They're often dispersed with the fouling communities attached to ship hulls or attached to aquaculture materials or other artificial structures such as buoys or marine debris. In addition, those globally distributed Ascidians have long breeding seasons, high reproductive outputs, and rapid growth rates. They can also tolerate a wide range of environmental conditions, including varying degrees of salinity, temperature, and Ascidians are also strong competitors for space and resources, and when introduced to a new location, they often outcompete our native species. Ascidians are so conspicuous that you can even find them in video games. But more importantly, they are the good guys helping save the world. Some Ascidians are also edible. Yum yum! Edible Ascidians are solitary forms that can be eaten with the tunic, like in this picture here with the tunic, or without the tunic, like in here. It depends on what you like better. They are consumed in several countries, including Korea, Japan, France, Russia, and Chile. In fact, they are so well-liked in Korea and Japan that they are actively farmed, like we farm tilapia in the US. Ascidians may be an acquired taste, but learn to like them because they are really good for you. They are high in protein and low in calories. Perfect combination. And if you learn to like them, even better for you, because not only they are a great food to eat, but eating them makes you younger. Eating sea squares reverse signs of cognitive decline and aging. So multi-million-dollar business here in the making. Ascidians are so great that they even produce something that we call marine natural products. Since Ascidians are sessile as adults, they can escape when something tries to eat them, or something grows over them, or something settles above on them. So what they do, they produce some chemical molecules called secondary metabolites, and they use those chemical molecules to fight against predators and competitors, etc. The cool thing is that those secondary metabolites can also be used by pharmaceutical companies to develop some treatments for us. So, for instance, in this case, in the slide, you can see an awesome sea squirt called Actinidea turbinata that is also right behind me. That's how awesome it is. And from this sea squirt, they isolated a molecule called ET743 that is now currently being used to help treating soft tissue sarcoma and also ovarian cancer. Yes, sea squirts. Like we have awesome microbes in our gut that will help us process our food, Ascidians also form symbiotic associations with a wide range of microbial symbionts. And this includes not only bacteria, but also archaea and also fungi. And we know that at least some of those microbial symbionts are involved in the carbon cycle, the nitrogen cycle, and as I just mentioned, in the production of those bioactive secondary metabolites. The presence of some of those symbionts, in particular cyanobacterial symbionts, those bacteria that have the chloroplasts, can also dictate the color of the colony. For instance, the green here in this Didymnum mole colony is dictated by the presence of a cyanobacteria called prochloron, while the pinkish color in this Tridymnum solidum is dictated by the presence of another symbiont called Synechocystis tridymni. But symbionts are not always responsible for the color of the Ascidian. Sometimes it's just pigments, different pigments like carotenoids, flavulins, or melanins. Or sometimes it's just due to the presence of spicules, those little calcareous structures that you can see in this picture here, and that will turn parts of the colony, like in this Didymnum duplicatum picture, white. Right? So this white in this case is due to the presence of spicules. Ascidians can have many colors, as you can see in these slides. Color differences are not only observed among species, but we also have some species that have different color morphs. So this is called intraspecies variability. For instance, this colonial Ascidian, Thistodites de la Cagey, here can be purple with white spots, or can be white with brown spots, or can be green. This is also true for other bigger solitary individuals, like this Polycarpa aurata, which can be yellow or can be purple. To make things more interesting, sometimes you can find different colors even within the same colony. Like this green pinkish colony of Lysoclinum fragile from the Bahamas, or this beautiful colonial species from the Philippines. Ascidians are filter feeders. This means that they're gonna get water in their inhalant siphon. As you can see here, we put a dye, so you can see how the animal is gonna get this water with the dye, and then it's gonna filter it in the body, and then it's gonna release the dye because it doesn't wanna eat dye. So it's gonna release the dye in a siphon that we call the excurrent siphon. If you bother the animal, he can close or open those siphons, as you can see right here. And then it just keeps releasing the water that was in the body. When an animal can do this, take food from the water column, we say that is a filter feeder. Let's talk about their anatomy. So here we have an Ascidian in purple, and if we look at this animal very carefully, you're going to see two little dots here within this circle, the red circle. If we were to dissect this animal and look what's inside, we will see something like this. Those two holes is what we've seen in the video before, the incurrent siphon and exhalant siphon. And inside there is something that we are going to see more carefully in the next slides that's called the thorax. That's where filtration happens. And under the thorax, we have something called the abdomen, and that's where we're going to find the guts, as in here, and in the stomach. All these together, the thorax plus the abdomen, is called a zooid. In Ascidians, we have different kinds of organizations. We have what we call colonial Ascidians, and in this case, the zooids are embedded in a shared tunic, like we just saw in the other slide. But sometimes the zooids are not all together in the same tunic. They are just attached at the very base by a stolon or a small tunic filament. Those Ascidians are also colonial, but to differentiate them from the others, we call them social. And finally, we have our solitary Ascidians that are formed by a single zooid. So it has its one zooid with its own tunic. It's not attached to anything or anybody else. The lifespan of a solitary Ascidian seems to range from several months to 2 to 3 years. Since solitary Ascidians are bigger, let's start with the easy part and take one of those big solitary Ascidians and look how they're made inside. So first, we need to open the Ascidian. And how do we do that? Well, we take our scissors, as you can see in this picture, through the inhalant siphon, and we cut all around pretty much until the base of the exhalant siphon. Or you can go all the way, if you wish, to the other siphon. So this is the correct way to open a squirt, a solitary Ascidian. So when we have a squirt like this one, we will say that the part where the siphons are called the anterior part of the squirt. So the part where the animal is attached to the substrate, we call it the posterior. Where we started our cut by the inhalant siphon all the way to the base, we call it the mid-ventral region. And what will be the mid-dorsal region? Well, the opposite one, where the exhalant siphon is all the way to the base. If we now remove the animal from the tunic, this is the animal with the tunic, and we cut all around here, and we remove the whole tunic in this case, we'll see something like this, like a big ball with different structures that are still inside the animal. This is called the mantle. We will have to open this big ball through the endostyle. So let me show you here where the endostyle is. It's right back there, this yellow thing here, and that's where you're going to be cutting with your scissors along the endostyle. Now, once you do that, you're going to be able to see what's inside the animal. So what you can see here is what we call the branchial sac. This is where filter feeding occurs. And the branchial sac will look different depending on which squirt species you have. In this case, we have a branchial sac with branchial folds, right? And we can use this to identify the animals. If we now zoom in one of these little sections of this branchial sac, we will see little squares here, little perforations called the stigmata. The stigmata, how many of them we have, how many longitudinal bars we have, how many transverse bars we have, all this is very important for us to identify the animal. In any case, the easiest way to see these structures is by coloring the animal with something called hematoxylin, like my colleague is going to be doing in the video.
So you just kind of let it go. It looks red initially, because when it's acidified, it's a little more red. But it'll turn blue and purple as we go. So just kind of get it out over the... And it's done.
Once we have observed everything that we needed to observe in the branchial sac, we can now carefully remove it so we can see what's hiding underneath. In this case, we can see structures like the digestive system. So here we have our gut, right? The gut, that will be the anus. And here we are going to have our stomach. In this case, a very long stomach, right? So very simple digestive gap for the sea squirts. So what will happen here is that all the particles, the food particles that are going to be attached into the branchial sac, it's going to be get attached by a mucus that the animal is going to push with the help of cilia, some little hair-like structures at the cellular level. It's going to be pushing all this mucus down to the gut. And that's where the digestion and then the absorption of nutrients will happen. Once everything is digested and the animal cannot get anything else out of there, the feces will be released through the anus. You can see in this slide right there, which is going to be taken those feces with the water that's being filtered, and everything will go out the excurrent siphon. Ascidians are hermaphroditic, which one or several gonads. So they have ovary and testes in the same individual. Ovary and testes may be juxtaposed, which means that they are one on top of the other, so close together that they look like one gonad. Or they may have many different gonads, like you can see in this slide. So in this slide, all these whitish things with GL on them are different gonads. And that depends on the species. So the number of gonads and the location of the gonads, they're all very important to identify your sea squirt. Gonads may mature simultaneously or successively. And the gametes that leave those gonads are going to be carried to the atrium right here by a gonoduct. You can see here the genital duct or gonoduct. The gametes are going to be released through this and out with the water again through the excurrent siphon. As I was explaining you, when those gametes are released in the seawater, they will be fertilized in the seawater. That we call an oviparous species, and that's what we'll see in most solitary species. But sometimes the eggs, the mature eggs, are held inside the parental body. In this case, the embryos are brooded until they complete the pre-larval development. When we have that, we call the species ovoviviparous, like many colonial species. The eggs will develop into swimming larvae that does not feed, and eventually it will settle for metamorphosis, normally very close to their parents. And once it goes through metamorphosis, as you can see here, then it will undergo a 90% rotation and it will open those siphons and start feeding. Once this happens, then the animal is sessile and cannot move anymore. Let's look at how a real larvae looks, in this case from a colonial Ascidian. You can see that it has a lot of different structures that we can observe for identification of our species. Again, those structures will be different from species to species. But in general, we have different numbers of adhesive papillae. You can see it here. In this case, we have 3. There is difference in the lateral ampullae. You can see them here very clearly. And here you can see the little zooid that is not filtering, but you can still see the structures, right? You can see an endostyle. You can see that branchial sac. You can see a gut here with an abdomen, and 2 different structures that the larvae will use to move around, something called the ocellus, and that will allow the larvae to know whether it's dark or if there is light, and something called the statolith, and that will help the larvae know if it's upside down or it's straight or what's going on. When the animal is in its larval stage, it can move, right? That's when the Ascidians can go from one place to another. Again, not very far normally, but some. And that's because they have a tail. You can see here the tail. It's still curled, of course, that will uncurl when the animal is ready to swim. And in this tail, we can see the notochord, right? A very important structure that in the vertebrates will originate the vertebra. So very important here. That's why we are all chordates. I hope I have convinced you of how important Ascidians or sea squirts are and that you've learned to like them or love them just like I do.